Second keynote speaker is Cedric Feschotte. So he was a PhD student in Paris where he worked on mosquitoes. And then he came to the US and did a postdoc with Sue Wessler, working on plant transposons, then got his first job in Texas, then transposed to Utah, and most recently to Cornell. He's used a masterful blend of computational and molecular biology to look at transposons of various types, most recently invertebrates. So it's a pleasure to have you.
Start the clock.
No, we're good. All right.
Okay. Don't start it. Okay. Yeah, so really happy to be here. What an honor. In the birthplace of Transposable Elements, I get to give a keynote. I'm still pinching myself. So I'm very happy. And this is going to be very different. By the way, I've never given this talk before, so I just put it together for this specially. And it's going to be much lighter than Josh. No data, almost no data today. True, almost. And now really what this is going to be a tribute to my mentors, to my trainees, and to many of my collaborators. So a lot of pictures, and so these are the people that have accompanied me on this path. As you can see, it's not a straight line. I'm not going to talk about the dead ends, just because I don't have time. But there were certainly many. And so, bear with me. We're going to walk through a lot of things together. Where is Sue? Trying to see. Oh, here she is. I'm going to talk about you in a minute. Okay. Yeah, so don't ask to explain the systems. So I'm not going to do that, because these are the systems we're working on. And there are many over the years. And actually, it's only some of the organisms that we've studied. We've mined transposons in the genome of many of these organisms. I put maize up there because I don't know if Sue remembers, but I actually did grow maize and cross maize mutants, the MOP1 mutants from Vicky Chandler. You remember that? To look if transposons were jumping around. Didn't work, so I'm not going to talk about it. It's one of these dead ends. Damon did something very similar, Damon Leash, with another transposon, and actually it worked. Anyway, so that's my contribution to maize transposon genetics. It was not much. Anyway, so the reason why we can study so many organisms, well, it's because first I'm a naturalist at heart. I love organisms, and I actually like to understand, or to try to understand, why a bat is different from a python, or a human different from a chimpanzee, which is a little bit harder to tease apart. And also because a lot of the work we do starts at the computer. And, my weapon of choice is actually, well, it used to be BLAST. BLAST is so great, right? You can play with it. I'm going to tell you a lot of BLAST stories tonight. And BLAST has no species boundaries. BLAST can go, if you don't restrict it, it would take you to many places. And indeed, I like to say that we go to where transposons take us. And as you know, they are everywhere, so potentially it could be everywhere, really. And really, but the point here is that by doing this, by studying many different transposons in many different species, I think we've gained really an appreciation of something very important, that probably my most important take-home message, but to this crowd it's going to be an easy one, is that transposons, they're not just cool, they're complicated. They're complex, they're different, so they all, in all these organisms, for the same business, because they're selfish genetic elements. They can replicate faster than the host cells that they parasitize. That's why they're there. We know that now. But they do so in many subtle ways. They have a lot of bells and whistles, and even two different line elements, two line 1 elements, even the two line 1 elements in the mouse and human are a little bit different. And that's the beauty of the transposon world, I think. And so indeed, it's possible to now view the genome as an ecosystem, teeming with transposons. These establish interactions, complex interaction with their host, as well with each other. And so indeed, it's not too far from an ecosystem. And in fact, this goes back, all the way back to my postdoc work with Sue. where we studied mites. Mites, miniature inverted repeat transposable elements. I'm sure Sue is going to talk about mites, so I'm just not going to get into a lot of details here. But they were very mysterious when I joined Sue's lab. They were discovered in Sue's lab by Tom Bureau in Mays in the early 90s, and then later in Rice. And the mystery about these elements is that they were very short. They had no coding capacity. They looked like DNA transposons, but they had copy numbers that we had never seen in DNA transposons, like AC and DS, the McClintock element, don't really reach these kind of copy numbers. Here they were in thousands of copies for a given family. And we were like, how do they do this? And where are the transposes? We really were just obsessed with this, Sue and I, with this question. And everyone in the lab at the time, we were chasing these transposes. And the way we went after that was to look in rice, because rice was the first complex plant genome that was being sequenced at the time available to us. And it was teeming with mites. There was 100,000 mites in the rice genome. So I delved into this for some time. And to make a long story short, came up kind of empty-handed actually at first. We knew I could guess what kind of transposes were moving some of these mites, but there was no direct relationship between the transposons, the long one that encodes the mites, and the short one, the mites themselves. You know, the tiers are similar, the target site duplication were the same. You know, we could implicate the transposes, but there was no direct relationship, and that really puzzled us for a while. And so I came up with this kind of model that, in fact, maybe they are moved by these elements through this sort of cross-mobilization going on, where the transposons encoded by an element actually moves elements that are not directly related to it. And maybe that's how they avoid silencing mechanisms or other features, and then maybe that's why we see them increase in so massive copy numbers. Okay, that was the idea. And it took actually 6 years for some of these models to become, I think, a bit more true. And I tried really hard to demonstrate this doing experiments in the bench, but I was not that good at that. But there was another postdoc in the lab at the time, and I joined the lab, Go-Jun Yang, that actually used yeast and set up a transposition assay where he could really dissect in great detail the relationship of these transposes, the biochemical activity of these transposes to the transposon, the mites. And indeed, what he found was indeed the mites move better when they use the transposes that's more distantly related to them. So that was, you know, so it's kind of a parasite of a parasite, right? So that fits with this idea of the ecology of the genome. Okay. Move forward a few years later, when I started my lab in Texas, my first graduate student, John Pace, so we moved to vertebrate genomes, to mammalian genomes, still looking at DNA transposons at first, because they were just not a lot had been done actually on these elements. I've seen Arian Smith here in the audience somewhere. Really, I was heavily influenced by Arian, who had described some of the first DNA transposons in the human genome. So we kind of walk in the footsteps of some of his work. But then John made a very interesting, I think at the time and still now, it was a stunning actually discovery to me, where he found some transposon, a transposon family, a hat DNA transposon, just a regular hat element, that was essentially identical between the genomes of all these organisms here, which are very distantly related to each other. And we see it, you know, in the Vespertilion bat. We don't see it in the fruit bat. There was a very patchy distribution, and the elements were essentially identical to each other. And there were thousands of these elements in these genomes. So we went on, and we, you know, a PCR to make sure they were not like some sort of contamination. And anyway, the inevitable conclusion or explanation for such a level of sequence similarity between so distantly related organisms was that these transposons must have come horizontally and infiltrated the germline of these organisms way after, of course, they diverged from a common ancestor. And in fact, John was able to date these infiltrations, these invasions, to about 20, 40 million years ago. Okay, so that, and actually, so it's not fair. I don't do any of this work, okay, not anymore. And yet I get to name the transposons. And so I named these transposons space invaders, and that's what I do, you know, I do the marketing. But actually, it actually worked. This is how I end up on a TV show with Morgan Freeman, actually. Just solely because we call these space invaders. I'll tell you more at the bar about this, but that's a true story. It's a bit embarrassing too, but I can tell you more. Okay, so then we moved on, and okay, no, before I move on, here at the time it seemed a little bit of an anecdote that these transposons would be moving between species as well. Now, there had been, of course, before us, some classic cases, the P element, Margaret Kidwell, was a classic example, but there was relatively few examples at the time. I think our space invader story was the first case of a DNA transposon transfer in mammals. But now, in 2018, it's a very different picture. In fact, there is a database out there maintained by Gabriel Wallo in Brazil. He called HTT database, where he curates manually, he goes to the literature and collect the strong cases of horizontal transfer of a transposon. And there's about 3,000 cases now. So in fact, it's not an exception, it's actually the rule. When you look at a transposon in the genome, you can almost assume that these elements, at some point in their history, have gone into this lineage horizontally. I think that would be fair to say now. Okay, well, of course, the big question has always been, how does this work? Do they really come from space? Actually, Morgan Freeman really thought they came from space. I need to tell you this. And when I told him, no, this was a joke, he was disappointed. But we still got on the show, so that was cool. But, okay, so how's that work? And I'm not going to give you an answer because probably there's many answers. That's what we think right now. There's many mechanisms and many facilitators of these transfers. And one that we think we didn't nail really, but we had some good hints for was host-parasite relationship. And that came from, actually, I studied this, actually. I blasted the spin sequence one day that we had found in mammals. And I got hits in this bug, but very good hits. Again, almost identical elements. This is a kissing bug. This is a blood-sucking insect. It's kind of gnarly. And in humans, they transmit trypanosomes that cause Chagas disease. And again, I remember that night, I was at a FACET meeting when I did that blast search, I remember. And I actually called Clément and Sarah, two great postdocs at the time in the lab. I said, hey, I found spin in the bug. I'm kidding, I'm not kidding, I found it. And they set themselves in motion. Immediately, they figured out a way to get DNA from this. We get extract DNA from the legs, not the gut, and all that, to make sure it was really in there, and da-da-da-da-da. And they also found 3 more families of transposons that were shared between a subset of the species that I've shown you earlier, some of these vertebrates, and the bug. So there was not just spin, there was actually 4 elements that were shared. And interestingly, when we made a phylogeny with the ancestral, the consensus sequence of these elements, as you can see, it's completely at odds with the species phylogeny. What we saw was very interesting is that there was a grouping of the elements by geography, by the geographical distribution, rather than their species proximity. And interestingly, the bug was identical, the consensus, 99%, I think, or more, to the possum element, the spin element. And so that's as close as a smoking gun, I think, although it's not a demonstration, for a potential direct transfer between a host and a parasite. Oh, what I didn't tell you is that these bugs, yeah, they do bite humans. But if you go on Wikipedia, which is the first thing I did when I found this thing in the bug, I'm not kidding, I go on Wikipedia, I look, what do they like to bite in nature? Possum, #1. No kidding. So I was like, wow, this smells really interesting. Okay, so by the way, also an interesting part of the story is that these elements were slightly older than these. So we could actually track that the movement, this transatlantic jump, had occurred in that direction as well, most likely. These were quite a bit younger, by 10 million years. So again, that's interesting because it shows you that somehow the ecology of the species shaped the ecology of the genome in a way. Okay. Okay, so then we moved on to another type of horizontal inheritance. These are viruses now, viruses that made their way into the genome. We call them endogenous viruses. They infect the germline and then become permanent fixture of the genome. And of course, this is very well described for retroviruses as well, of course. And but we got excited this was in 2008 actually, when Clément, who had actually done some field work in Madagascar, it turns out, he did his PhD in South Africa. Clément had some nice samples of these rare Malagasy lemurs in the freezer. And one day I actually found, I blasted again, sitting on the couch one night. I was taking HIV sequence. I was like, hey, let's look for an endogenous lentivirus, because this had not been seen before. And I found one in the Microcebus murinus genome, 1 copy only. So I told Clément the next day, hey, Clément, let's get on this. This is interesting. He's like, yeah, I got samples in the freezers of these lemurs from different lineages. So he did experiments, he did PCR, he did southern blots. Yeah, we did southern blots. And to show that, in fact, there was not just one infiltration of these lentiviruses, but two independent infiltration about 5 million years ago in two different lemurs. That was really interesting to us. This was the first primate endogenous lentivirus. And another interesting discovery that he made was to find these not retroviruses, but HEPA-DNA viruses, things related to HBV, hepatitis B virus, in the genome of these songbirds. So this was a little weirder, because these guys are not supposed to integrate in the chromosome as retroviruses do. Yet they did, and they did that in the germline, since they got fixed now in these genomes, and we could infer that they were 20 million years old. And this was also very surprising to virologists, because if you take, and that's the sort of the take-home of this kind of paleovirological studies, you get now access to very old viral fossils. And that gives you really unique insight into how viruses evolved on the long term, which was essentially inaccessible without this kind of fossils, because if you look at viruses that are circulating now in the population or in different organisms, they evolve very rapidly, and you lose track of their evolution very quickly. So if you ask, before the studies, someone studying the evolution of HEPA-DNA viruses, the deepest they would have come up with for the emergence of this group of viruses was a few 100,000 years. Now we were telling, no, these things have been with vertebrates for a long time. Okay. So again, and more recently, actually, a graduate student, former graduate student in the lab, Shou Zhou, found another really cool case of transfer of endogenous retroviruses, that this time essentially the same retrovirus infected bats, pangolins, and the ancestor of cats about 10 million years ago. Okay? And this was surprising, because actually there hasn't been a lot of long-range transmission of retroviruses described. You're probably familiar with the fact that HIV we got from chimpanzees, but this is our closest relative, right? And it's often thought that phylogenetic distance, phylogeny sort of constrain the movement of retroviruses, but clearly some retroviruses are able to go between species. So anyway, all of these studies, I think, are interesting because they tell us two things. First, they tell us that the genome has incredibly low boundaries, low borders, right? It's really a grab bag of stuff that you can acquire from the environment. So that's an interesting concept. It's very clear, it's very well established in prokaryotes, but I think in eukaryotes it's still very underappreciated. And another thing that we learned from this type of studies is that, once again, we can learn by looking at the genome, we have a record of interactions between species in the distant past, right? Which I think is very, very interesting and fascinating for me, at least as an evolutionary biologist. Okay, now I'm going to shift gears completely and tell you a little bit about what happened once these elements established themselves in the genome, spread, and then what else? What do they do? And here really, the type of impact that we are interested in is more about a long-term impact. We're going to hear a lot of talks at the meeting about the short-term impact of transposition, in particular as insertion or mutagen. We're interested in something else. This idea that I think it was Wojciech Michalowski in the 90s that came up with this idea of genomic scrapyard. This is the idea that this material accumulates, slowly decays, especially in mammalian genomes, and sits there, but on occasion, can provide the raw material, substrate, for new evolutionary novelty to come up. And this can happen very quickly after insertion, or later on, long after insertion. Okay, so it's like repurposing, recycling, or co-option of transposable elements. And really, also, we are really heavily influenced by another paper that you need to take a look at if you haven't yet. It's a fascinating read. It's a really fun read by François Jacob in 1977. And it's the idea of tinkering. It's the idea, which actually Darwin already mentioned early on, that in evolution, probably nothing really comes from scratch, from completely novel things. It's mostly going to be a process by which you recycle and you reassemble parts, existing parts, to come up with novelty, okay? And that's what François Jacob talks about in this paper. And again, he says, without any well-defined long-term project, So this is very important. There is no anticipation here. We're not keeping this stuff because we know it's going to be useful later, but it's just there's no defined project. The tinkerer, when he says the tinkerer, he's not talking about some sort of creator. He's talking about natural selection. Gives these materials unexpected functions to produce a new object. So this is exactly the kind of things we're looking for, but with transposons, because we think transposons are great for tinkering. Why? Because they are complicated. They have lots of bells and whistles built in. Why? Because they've been selected to do their selfish business. So they do have cis-regulatory sequences, enhancers, sometimes insulators, sometimes other promoters, of course, to drive their own gene, which are exquisitely specific. We know now that. They make RNA, coding, non-coding. They make many proteins, many activities. So there is a lot of things to play with here. Okay. And then, so I'm going to give you two flavors of co-option that we've been working on. First, the non-coding kind of regulatory types. Think about transcription factor binding sites or enhancers derived from transposons, and then the coding sequences. A little bit, a few vignettes, no data. Okay, so the idea of controlling, of course, transposons controlling gene expression is not new. This was discovered here on this campus. Is the idea of McClintock of controlling elements. That's how she called these elements. She didn't never call them transposable elements. She called them controlling elements. Okay, and indeed, she thought at the time in the 40s and the 50s that this was how genes are regulated in eukaryotic cells. Okay, this is even prior to the lac operant of Jacob and Monod in the 60s. We know nothing about how genes are turned on and off during development. And when she made all this observation with AC and DS first, and then later with SPM, she really thought this is how genes are regulated. Okay? We know now that probably she was a little off in the sense that that's not why transposons are there. But as we're going to see in this meeting, this idea have come back in a sense that yes, transposon can sometimes regulate adjacent genes. And I need to say also another source of inspiration was the work of Sue first in the 80s with mutants of maize, but also later with what looked like ancient transposon or remnants of transposon that they had observed, and Tom Bureau had done a lot of work there, near genes that appear to provide transcription factor binding sites or polyase signals and things like that. So really, these were pioneering ideas. And another one was the Britton-Davidson model of gene regulation, came up in 1969. So again, here we're trying to solve the problem. This is after the lac operant, right? So, but here the problem in eukaryotic cells is that we have genes broken up into different chromosomes. So how do you coordinate a gene battery? Genes that need to be turned on and off in response to an environmental cue, or what we think now, for example, is a transcription factor. And their idea was built upon their own discoveries that eukaryotic genomes, including the human genome, were incredibly complex and full of repeats, of interspersed repeats. At the time, no DNA sequencing whatsoever. This was all inferred from reassociation kinetics, okay? And they knew that they were, if you go back to the 1968 paper from Cornell and Britton in Science, this is amazing. They were able to infer, Ariane would appreciate, I'm sure you remember this paper, that there were hundreds of families of transposons of different age of decays in the human genome, which is, we know now is true. That was just from reassociation kinetics. Anyway, so they were well aware of the structure of the genome, and they thought, well, maybe some of these repeats from the same family can be how genes are wired. Sorry, I'm trying to point with this. Sorry. And indeed, if they share the same copies of the same family of repeats, then you can turn them on or off at the same time. Okay, so this model has gained a lot of popularity lately, especially with the advent of functional genomics and regulatory genomics. Now, the ability that we have, in a fairly unbiased way, although there's still some biases, we're actually still not seeing all the repeats, to map the binding in the regulatory activity of the genome, sort of a genome-wide, okay, including transcription factors. And this is a study that was done from using ENCODE data some years ago by Ting Wang's group, which is a very nice study, where they looked at about 100 different human or mouse transcription factors in a variety of cell types and cell lines and tissues. And basically, any transcription factor you look at, there is a substantial amount of binding in transposon. Well, you're going to be like, well, yeah, sure, they make a lot of the genome. We know that. It should be some by chance alone. But the key point, the salient point here is that for a given transcription factor, most of binding events in transposons is dominated, enriched for one or few transposon families. And then when you look at the transposon, you see the binding site for the transcription factor, and you see it in the consensus ancestral sequence, which means it's consistent with this idea of the Britton-Davidson model that it was there prior to the spread of the element. The element spread the binding site, and now we see a lot of elements that are still bound in the genome. But of course, that's not telling you they're regulating genes. Some of these studies got to this in a correlative way, comparing binding events to activity levels of genes that were adjacent. But again, the big question is, does it really matter? Oh, and I need to say, another view of this is the transcription of the genome, this sort of pervasive transcription. Well, pervasive transcription is not random transcription, okay? Here we're talking long non-coding RNAs, and you know in the human genome we have about 20,000 or more by now loci, genes, that have a promoter, a transcription termination signal, multi-exons. We're only looking at the multi-exonic, so these splice sites and so on. They look like normal genes, but they don't have any coding capacity. And Aurélie Capusta, a postdoc in the lab, realized that actually a lot of this transcriptional activity, a lot of these promoters for these link RNAs come from transposable elements. And I should say almost all of them come from the long terminal repeats of endogenous retroviruses. So not every transposon, again, a biased set of elements. And they are developmentally regulated, cell type specific, and of great interest to us, they are often species or lineage specific. Most of those will be, in the human genome, will be primate specific. So if they are doing anything, they will be doing something that's lineage specific innovation. Of course, once again, the big question is how much of this really truly matters to the organisms. And this is really what we want to get at, and what we've been working on in the last 5 years, is to kind of move from this sort of descriptive view of the genome where we can map regulatory activities. Sometimes you can associate this with gene expression to really trying to see phenotypes. If you can manipulate, for example, delete the element, then you should be able to see that indeed this gene is regulated by this transposon, and hopefully what we would want to see is that there is a consequence for the fitness of the organism if you, for example, delete or mutate the element. Okay, so we've been attacking this question from many fronts, and many people in the audience. I've came here with 6 people from my lab, so I hope you're all going to see their posters. I'm going to give you a few vignettes of what they have to present. And really, one of the key here is that we're basically going after biological systems that we think are physiologically important and interesting, where hopefully if we manipulate the elements, we can actually see phenotypes, and often systems that are tractable in cell culture. Okay, so we've been looking at development in collaboration with Gunther Wagner and Vinnie Lynch. We've been looking at the evolution of pregnancy. Tommy Carter at the meeting as a poster on embryonic stem cells. And we are interested in looking at what we think is a regulatory switch between proviral LTRs and solitary LTRs. Turns out they have different regulatory activities. Tommy is looking at that. Sylvia here, is focusing on zebrafish. Yeah, this is a fish. I know, it looks funny, right? This is an embryo. And here, what's cool about fish is like the embryo is very accessible. So if you want to manipulate transposon early in embryonic development, we think this is the system to go after. So check out our poster. She's analyzing single-cell RNA-seq data now for the zebrafish embryo, and it's turning quite interesting. Ed is, and in collaboration with Nels, LD has focused on innate immunity. I'm going to tell you just a little bit more about this in a minute. Julius here has a poster on enhancers that are circadian enhancers in the mouse liver. So this is a cool project that was initiated by an undergraduate about 4 or 5 years ago now, which Julius is taking to the next step. And then we also have Clément at the meeting, who has a poster on now the more like short-term impact of transposon on gene regulation. And Clément is looking at how polymorphic elements in humans affect gene expression variation. Okay, but really another really game changer for us was brought by this guy, Ed, to my lab, was CRISPR, right? It is really a revolution for us working on transposons, because we can indeed knock out virtually any element in the genome. And even us could do this. So I think a lot of people can do this. But now, Ed brought this to the lab. He came 5 years ago, he came to the lab, he was like, hey, let's CRISPR these guys. And this is what you want, right? You want someone that's fearless like this and does the experiment. That's what he did. And in one study we published, which I really won't detail, he used guide RNAs, specific guide RNAs, to delete individual elements and create homozygote deletion cell lines for elements we suspected regulate important gene in the innate immune system. And he did that, and in every case, I'm showing you all the data we generated at the time. Of course, we handpicked these genes because they were interesting innate immunity genes, and in every case, we could show that the elements really have a substantial impact on, in this case, this is interferon inducibility of the gene upon infection or upon interferon treatment. So indeed, they do something to these genes. And in one case, we were able to go down to the phenotype and show that the mutant cells are more susceptible to pox virus infection than the wild type cells. So this was a really more of a true cellular phenotype. Okay. Okay, so this is looking a little bit more like a Britton-Davidson model. And of course, now we can scale this up. And in fact, Ed has started his lab at the University of Colorado Boulder, and he's doing that in his lab. We're going to do different screens in our lab, but you can now test these on a very large scale using lentiviral delivery systems to do pooled CRISPR screens. So that's where we're moving. Okay, I think I've got a few minutes, couple minutes to tell you, 5 actually, good. Tell you a little bit about coding sequences now that are recycled from transposons. So we've been working on this for a long time. Many different cool stories. So this is the story right now. You got a transposon genes that go to the from the dark side to the bright side. It become a respectable gene that you can put on the pile of functional genes. This is a gene under purifying selection. We can infer this, and now it's annotated as a normal gene. And often these genes have no longer the features of transposons, and usually they are immobile, right? They're no longer able to move around. So we've been studying this for a long time, and the cool part about this is that it's going to take you all over the place, all over the cell, because any activities of these transposon genes, protein-coding genes, can be recycled for cell function. RNA binding, DNA binding, many transposon proteins do that, packaging as well. Catalytic activities, I think we're going to have a couple of talks on VDJ recombination and RAG1 and RAG2, fantastic example of that. I think Alex Kensis, I don't know if he's in the room, is going to talk about PGBD5. Alex and I actually have been collaborating on this for a long time. We think this is a RAG1 kind of story in the brain, so we're excited about this. And interaction with host proteins, I'll show you just one example of that with envelope proteins that binds to receptors of host cells. So any one of these activities can be recycled. And one we kind of stumbled on lately in collaboration with Jason Shepard at Utah was this ARC protein that was known to be involved in memory formation in the brain, in the mouse brain, in the murine brain, but through very unknown and clear molecular mechanisms. We know it's expressed at the synapse. We know it stimulates the turnover of glutamine receptors, but at the molecular level, it was very unclear what this ARC protein does. It was known that it was related to a capsid, a gag protein of a retrotransposon or retrovirus, because when they made a structure in 2015 of a partial structure, crystal structure of ARC, it was immediately clear it could be superimposed to the capsid of HIV almost perfectly. And basically in this study that was published earlier this year, Jason's group was able to show in neuronal cell culture and also in vitro with biochemical experiments that, in fact, not only looks like a capsid, but it does exactly what a capsid does. It binds, it packages its own RNA, the ARC RNA, and these capsids are transferred across the synapse, at least in cell culture. And then Vivian Budnick, working completely independently in flies at the neuromuscular junction, found that also the Drosophila ARC homologue does the same thing at the neuromuscular junction, packages RNA, and she was able to go even a step further and showing that this trafficking of this RNA across the synapse was actually important for synaptic plasticity in the fly and memory formation. But remarkably, what we found, John McCormick, a graduate student in the lab, realized that these were domesticated twice, not in the common ancestor of flies and vertebrates, but in fact, we could show that the ARC in mouse goes back to the tetrapod ancestor, so about 300 million years ago, and is derived from a gypsy element. But the Drosophila ARC goes back to only about 100 million years ago. It's not in mosquitoes, it's not in beetles, it's not in butterflies. And it comes and it's most closely related to gypsy elements, different gypsy elements in these lineages. So again, this is kind of a case of conversion, co-option of a gag protein for similar cellular function, and here it looks like a completely novel mechanism of RNA delivery at the synapse. Okay, I'm almost done. Transposons. We're going to hear at the meeting about RAG1. I'm not going to tell you about these. PAX6, spectacular example. This is essential for eye development. The pair domain of PAX6 come from transposase. Many years ago now, we worked with Haeyang Wang, which was at Cornell at the time, on a story where a mutator transposase has been transformed into a transcription factor. This is a standalone mutator transposase that now controls light sensing in Arabidopsis. So spectacular example from the biology standpoint. But the issue with this, and the question we've always had is, how does it work? How do you start to domesticate this transposase? And for this, we turn to the bats. Why? Because we've learned from work we've done with Ellen Prudham, Nancy Craig, David Ray, that bats are really interesting for DNA transposons. Turns out like no other mammals so far that we've looked at, they are the only ones that have a lot of DNA transposons, very recently active, and in fact, Nancy and I, we were able to show with piggy bat that some of these elements are still currently active. So then Rachel turned to bats, and Rachel has a talk on Sunday, so I'm not going to steal her thunder, just a little bit here, but not too much. And she's been working on the bat-specific crab transposase fusions. We're going to be about crab zinc finger genes from Didier Trono in a few minutes. So here we think that these transposase fusions are transcriptional repressors that in fact evolved first to repress the cognate transposons. And we think this is the stepping stone for this transposase, crab transposase, to become transcriptional regulator in development and other functions. Okay, and she found a lot of those all over the place. 30 independent crab transposase fusions. So it's a recurrent theme in evolution. All right, the very last bit is a story that John Frank in the lab is working on now for a couple of years now, has to do with sensitin. And again, this is a spectacular example of transposon domestication, mostly documented from Thierry Heideman's group in France. And Thierry's group has been able to show that in many mammal lineages, you have different retroviral envelopes that are expressed in the placenta that are fusogenic, which he proposed are involved in the formation of a syncytial trophoblast at the interface between the mother and the fetus. This is where there is cell-cell fusion. We have a little bit of a different take on this. John is actually testing not a mutually exclusive possibility, but a different idea that, in fact, what promotes, at least initially, the domestication of the syncytin, these envelope proteins, is actually antiviral defense, simply because they will block the receptors that exogenous retroviruses can no longer use. And so John has some exciting data actually showing that this is probably true for syncytin 1, at least in humans. All right, I'm going to wrap it up and tell you, just summarize some of the things we've learned along this road, this meandering. So first, yes, the genome is an ecosystem teeming with transposons, a lot of them, many different types. That's the beauty of it. They have no borders and no shame. Indeed, that just means that, yes, they get in the genome, they spread whether you like it or not. And then evolution is mostly a matter of tinkering. We know that you don't really make brand new things from scratch. And we think transposons are great for tinkering. That's all I wanted to say. These are the people that really do all the work, the funding, and I'll be happy to take any questions. Thank you. If you have. You never know if it's good or bad when people have no questions. Mostly bad, huh? Right? I mean, absolutely.
It's perfectly clear. All right, so I guess we'll take a break.